Bueno, buenas tardes a todos. Bienvenido a los 17º Coloquios de Microbiología Valparaíso 2020. En esta ocasión tengo el privilegio y el honor de introducir a la doctora Fujiyoshi de Japón. Es el primer coloquio de Valparaíso que acoge a una investigadora de Asia. Fujiyoshi estudió biología en la Universidad de Hokkaido, en Sapporo. A continuación, realizó sus estudios doctorales de agricultura en la Universidad de Kioto, en la ciudad de Kioto, donde posteriormente fue profesor asistente. Actualmente es profesor asistente en la Universidad de Hiroshima, en la ciudad de Hiroshima, Japón, una ciudad que solo tiene 2,8 millones de habitantes. Ella trabaja en colaboración con Chile desde el año 2018, cuando empezó un proyecto llamado SATREPS, en colaboración con el profesor Dr. Milko Jorquera de la Universidad de la Frontera en Temuco, en el cual también está involucrado el profesor Fumito Maruyama. Y sus líneas de investigación son la microbiología ambiental, las interacciones microbio-microbio, la genómica microbiana, y ella nos va a hablar hoy día en particular sobre la caracterización, characteristics of the microbial community in bioaerosol in Chile and Japan. So thank you very much for this colloquium that we're waiting, and we're very happy that you can join this colloquium of Valparaíso. Thank you.
Thank you. Buenas tardes. ¿Cómo están? Estoy feliz de tener esta oportunidad de hablar con ustedes hoy. Honestamente, el mes pasado no pensaba que este coloquio se realizara debido a esta terrible situación. Pero gracias al profesor Mijael y a todos los interesados, estoy aquí. Más allá de la diferencia horaria de 3 horas. Esto. 3 horas. Estamos conectados ahora. Y me gustaría expresar mi gratitud al profesor Mijael, a la doctora Vanessa y a todos los interesados. Antes de la presentación, permítanme presentarles brevemente a Japón. Desde aquí hablo inglés. In Japan, this is a Japan photo. This is taken by Mijael. He visited Japan this February. He takes a photo. And this is a typical Japanese photo. A lot of terrain, complicated metro map, a lot of people. And the population in Japan is about 126 million people. It's about 10% of Japan called Chile. And Japan land size is half of Chile. This is Japan, and this is Chile. And Tokyo, capital of Japan, is 9.2 million people. There's a lot of people. And in the morning, there's a lot of people coming to the station and go to the company or workplace. So every morning you can see this kind of people, a lot of people. But this is one side of the Japan because the human landscape appears so overcrowded in the some part like Tokyo, like this. But in some part of Japan is yet underpopulation in others like this or this. This is Tokyo, so very a lot of people. But this is Kyoto. This is Saijo, Hiroshima area. Not so much people, and that means a variety of lifestyle we can spend in Japan. And by the way, Hiroshima is Hiroshima is a very little city than smaller city than Tokyo. So 2.8 million people here. So not so much crowded and so much affected by corona infection. And today's my title is about bioaerosol. And why bioaerosol is very important is like this. Bioaerosol is a biological aerosol. It's a subcategory of particles released from terrestrial and marine ecosystem into the atmosphere. And both living and non-living components including. So some algae or archaea or bacteria, fungi, spores, plankton, a lot of kind of living or non-living components including. And from medical point of view, we inhaled respiratory rate per day is 28,800 times per day. And the average ventilation volume per session is about 0.5 liter. It's a 15 kilogram person average. So it means we inhaled the respiratory about 14,400 liters per day. We take in the air in my body, in our body. If this in this air, if it contains some pathogenic bacteria or pathogenic viruses, it's very infected human easily. Or sometimes it causes infectious diseases or allergic diseases. And the ecological point of view really play a vital role in the Earth system. From the forest or soil, its emission, and sometimes it's called cloud processing or transport or transmission and deposition to the other area. So microorganisms can spread and interact each other via air. So it's both very interesting from medical point of view and the ecological point of view. And this is my review publication. I published this in 2017 in Frontiers of Microbiology. Its title is a transmission of airborne bacteria across built environment. It's mainly focused on the built environment. At this moment, people in all over the world is very interested in this review, thankfully. Today, I checked my total review and it said 11,155 views. And mainly it was seen in the United States, and Chile is 65 view, thank you. And Japan is more less than the United States, 221 CC. But all over the world, the country are really interesting in this title or this research area. And let me briefly introduce my review. The main focus of this review is, as I introduced in the main focus is the airborne bacterial community is affected the human, and the human also affected to the airborne microbacterial community. The human health is probably, not sure, but probably affected by bacteria in built environment because people spend approximately 90%. I think at this moment it's more, or 100 or 99% of their lives there. So human and outdoor air are likely to be the major source of airborne bacteria as well as bacteria growing indoors. So this is open air bacteria also affected the airborne bacterial community, and bacteria growing in air indoor is also affecting this community. So this is like a cycle, also affected, human affected. So maybe this is a really tight connection to human health, to the airborne bacterial community in the built environment. So to know the built environment or airborne microbe, the source of the airborne source is need to be studied. And this is a typical source of airborne bacteria in the house. I separated 3 major sources shown here. Mainly, one is a human occupants, like a bedroom or living room, and the other is water, as like a bathroom or kitchen or wall, or other is outdoor, from the window or door or air conditioning system. And this review, what is the main focus on this review is I want to introduce this term. This term I want to show you, because last slide I show you that outdoor microbes also influence the house microbial community. So house and the community place and the open air is very connected. So I want to coordinate all of these total environment. These airborne microbes are easily transmitted, because this is I want to study as a meta community. This meta community term is firstly published in 2016. Limpero Polo, the researcher in University of California, adopted this meta community to describe the microbial composition of the indoor air, and which is influenced by microorganisms from environments surrounding like this, and the space in question. Therefore, the living space, neighborhood, and the whole city can be regarded as an ecosystem. So we need to, at this moment, especially at this moment, we need to study as a meta community of all these house and the community place and the open air. And based on this idea, we are now studying house, inside the house. We study the showers as a source of biology indoors. So we do just in Japón. We study in this bacterial community, and also we're studying mountain, compare mountain and the city. And also we compare suburban area and urban area. Thank you for, luckily, we just accepted this publication, scientific report, July 22nd. And today I also want to show you that the Chilean bacterial community from Osorno. It's a very briefly introduction, really light data, but it's very interesting data we can get. And also we studying in Punta Arenas here. So I want to show you this data. So today's talking take-home message. I think you already in home, so remember home message. I'm not sure, but this is a take-home message. This microbial communities mirror the environment. This is the take-home message of this presentation. So let's move on the Japanese bathroom. Why Japanese bathroom? Japanese bathroom is a very specific area, and especially for elderly people. Let me show firstly this slide. Japanese style bathroom or bath time is like this. We have hot water place and the washing space, and we have also shower. We also have the one, but the important point is separated from toilet. And we wash our body in this washing space, and taking a bath a long time, and taking also shower. And many Japanese people prefer to take a really long bath, more than 30 minutes every day. So bathroom is there is a lot of bacteria or microorganisms living here. So Japanese people maybe or probably highly exposed to these microorganisms. And the problem is this. Sometimes microbes in our home environment cause infectious diseases. It's very famous one is this one. Non-tuberculosis mycobacterium. It's called NTM. This NTM is living in soil or liver. So it is difficult to where they come from, but the patient, NTM patient, NTM patient house, there's an NTM in their shower head or bathtub inlet. There is a NTM we detected. So maybe during the taking a bath, they infected NTM at this time, bathroom time. So we are studying the microbial community in the shower head, and also this shower water. We studying of that microbial community. This is a brief result, and we compared our alpha diversity from the biofilm from the shower head and the shower head feed water. And more variety of bacteria are present in the water. This is alpha diversity CHO1, and this one shower channel index. And as you can see here, shower head is lower diversity than water sample. And also looking at the beta diversity, more water sample is more closely related to the other one. And in the other hand, shower head biofilm is a lot of diversity, each sample. So how different is the microbial composition in one environment compared to another? And we're searching for this. The maybe the relationship between biofilm and the shower water is like this style. The bacterial community in water were distinct from those in biofilms. For example, biofilm bacteria is mainly Sphingomonas and Methylobacterium, but shower head feed water is mainly Sphingomonas or Phragobacter, Methylobacterium, Ochrobacterium. Is a total 14 genera is commonly detected. So, and also compared with the beta diversity result, the proportion of the number of shared bacteria found in both water and the biofilm were about 2 times higher in the biofilm than those in water. So from these results, this may indicate that bacteria in water primarily adhere to the shower head surfaces and form unique bacterial community. So we also checked that pathogenic bacteria in this shower head water and shower head biofilm and the feed water, and Japanese shower room or bathroom is relatively safer than United States or Europe. Here is a NTM percentage of the United States and Europe. It's about in the United States, it's about 15% of the NTM, and or Europe is of about 5%. But looking at the Japanese bathrooms, mycobacterium is relatively lower than those in the United States or Europe country. Some shower head feed water is higher. So we need why this sample is higher, or this is why it's area, or it's seasonal, or it's lifestyle. We have no idea at this moment. So we keep sampling and we are looking for that what kind of factor is affected, this kind of percentage, pathogenic bacterial percentage is affected. Okay, so from this, we move on the airborne microbial communities, the Japanese city. So firstly, I want to introduce you that the mountain area, high altitude area, and the suburban area bioaerosol composition, microbial composition. So one site we taking from the sample is Toyama University. It's Toyama City, and just 40 kilometers apart from the Toyama University, there is a high altitude mountain. It's about 2,839 meters above mean sea level. It's just 40 kilometers different. And this is a mount called Mount Judo. It's a Tateyama Mountain. And also the other is Toyama City. This is Toyama City. It's not so urbanized area, it's a suburban area. We compare this microbial composition to see how different the two sites. And we sampled the bioaerosol on the rooftop of the Toyama University here. And the other area, Mount Judo. And the air samples were collected at the five time points from 2009 to 2011 on August or September. And the air samples were collected at the flow rate of 5 liters per minute over 3 hours. So mostly volumes, the total volume of the air is 900 per sample. And this sampling scene, we used a 0.4 micrometer polycarbonate filter. And we analyzed 18S ribosomal RNA, eukaryotic community composition, and 16S ribosomal RNA for prokaryotic composition. Okay, this is just show you that the bacterial community, taxonomical composition of bacteria and the eukaryotic community at the class level in the air sample. This is a little bit complicated, so I want to show you like this style. And mainly bacterial community, major bacteria is actinobacteria and alpha proteobacteria and gamma proteobacteria, bacillus, and the beta proteobacteria. This is really typical result, and the bacterial community congruent with those reported in the other study. And also we checked that the hierarchical clustering of bacterial community, the red one is sampled from high altitude area, and the green one is a suburban area. As you can see here, it's a mixed. So not so much different detected at the bacterial community. On the other hand, this eukaryotic community, this shows that this is this result is eukaryotic community. The major bacteria is Agaricomycetes, Dochideomycetes, and Magnoliopsida, the Ascomycetes, and Solidariomycetes. This result is remarkably similar to previous work, but difference in the eukaryotic community composition between high altitude and suburban site were observed. As you can see here, here is a high altitude areas eukaryotic community, and this green one is a suburban area. It's very clearly separated. So maybe suburban area and high altitude area has a specific eukaryotic community it has. And seeing this one, I think next slide is more easy to understand. This one, we do a linear discriminant analysis effect size. It's called the NEFSE method for biomarker discovery. Was used to determine specific eukaryote taxa in both locations. Blue one is a suburban area specific eukaryotes, and the red one shows that specific eukaryotes found it in the high altitude. The more abundant taxa in suburban it's Agaricomycetes, high altitude samples. This one, yeah. High altitude, yeah, this one. Sorry, this one is more abundant in high altitude sample, and on the other hand, Ascomycota was more abundant at the suburban site sample. And looking at this, what kind of eukaryotes of this is like this. This is Agaricomycetes. It's a fungi. This is more abundant than the high altitude site, and the Agaricomycetes is a mushroom-forming fungi. And on the other hand, more abundant than the suburban site is Dochideomycetes. Is like this. Dochideomycetes is the largest and the most diverse class of Ascomycetes fungi. They grow on woody debris, decaying leaves, or dung. So it's really easy to understand that both of these classes is abundant at each site. So this study's conclusion is bioreservoir has a locality only 40 kilometers different. High altitude site and the lower suburban site is a different microbial composition, especially in eukaryotic community. And also we compare that urban site and the suburban site. One is Yokohama city, is very urban site, a lot of people living there. And on the other hand, Toyama city is mainly mountain and a really great nature. And it's famous for great nature in Toyama. This is this year published one in scientific reports. And also it shows that clearly different microbial, this time is a bacteria, the community. The one background is to study size-related characteristics of airborne bacterial community composition, diversity, and abundance in each sampling site here, Toyama and Yokohama. We want to compare what is different from urban site and the suburban site, and also the morbidity of original in terms of prefecture is the highest in the country, and this trend has continued since 2005. So we want to detect Legionella from bioaerosol samples. The sampling site like this, Toyama City is Yokohama City is different about 360 kilometers, 400 kilometers is Santiago to San Jose. It's not so much different distance, big different distance. And the sampling tool we use for size resolved sampling, we use this Anderson air sampler. This has totally 8 stages. One is over 11 micrometer particle size. 2 smaller is this backup stage, smaller than 0.43 micrometer size. We can separate it, the particle size at each stage. And air sample, we use the quartz fiber filters and the flow rate of 28.3 liters per minute, and we take in over 24 hours. And we use the DNA extraction like this, and also we analyze the 16S ribosomal RNA gene or summarize it. This is a microbial bacterial composition. This is too difficult to see that, but this result was congruent with those reported in other biological studies. So it's not so much specific bacterial composition here, suburban or urban site. But what is interesting in this study is this. One is this. We compare the Shannon alpha diversity index of bacteria associated with the suburban sample. And looking at this graph, suburban sample is both greater than those associated with urban sample at both over 1.1 micrometer and smaller than 1.1. Sorry for cutting this. This one is cut it. But this one, so next, no. This red one, I think, this one is from suburban site, over 1.1 micrometer particles, and the green one, I think suburban site, smaller than 1.1 micrometer. And this one, blue one, is urban site, over than 1.1 micrometer particle. And this purple one is urban site, smaller than 1.1 micrometer. Let me see next. Yeah, it's corrected. Okay, so we also compare the principal coordinate plots and also hierarchical clustering bacterial community. And seeing at this one, principal coordinate plots showing that overall variation in bacterial communities. Community are compared using a Curtis distance, and it showed that suburban area, this red one, over 1.5 bigger size particle, and Yokohama area is urbanized areas, or smaller than 1.1 micrometer particle is totally different. And also seeing that the hierarchical clustering bacterial community, air sample from suburban site. Suburban site is suburban site is a green one. This one, green one is grouped separately from suburban. This red one and the urban site blue one is clearly separated. So this separate point, looking at this stage, this is the Anderson 4-stage area. This is Anderson. So closely looking at this stage, we can notice that this is clearly separated above or less than 1.1 micrometer particle size. And also, what kind of bacteria is specific for suburban site or urban site over 1.1 micrometer particle or below 1.1 micrometer particle. We are looking at this, we are using this Lefse again and identify the most differentially abundance taxa within 4 sample groups. And this Lefse show that high abundance of human skin sample, human skin associated bacteria, such as this one is urban one, such as Propionibacterium here, or Corynebacterium here, is more abundant at the urban site. And this maybe or could be indicate that outdoor bacterial community were influenced by bacteria from human associated bacteria. On the other hand, that suburban site, this red one and the green one, show that mainly Methylobacterium or Sphingomonas is associated with soil and plant microbiome. So this is from mainly natural environment, some bacteria. And this is a lot of differences between suburban site and the urban site. Also, it different over 1.1 micrometer particle size or below 1.1 micrometer particle size. This is a really interesting point of this study. Also, as I show you that Toyama area is very highly infected by Legionella Legionella, and we can successfully detect the Legionella from the particle size over 2.1 gross particles. This red one shows the sequence from cooling tower water sample in Japan. This one and this one, and the blue one is sequences from this study. It's very close to the water cooling tower Legionella sample, Legionella gene sequence. So, at this study is fastly detected. We can successfully and fastly detect it from bioresorb. We can detect it Legionella from bioresorbs. So this study's conclusion is that size resolve differences occurred in terms of airborne bacterial community composition and diversity at the suburban site area and the urban site. And the threshold of the particle size is over or below 1.1 micrometer diameter. At this from this study, before this study, usually samples or collected particulate matter at the PM 2.5, but for bacterial composition, we recommended that 1.1 micrometer of particle size is good for airborne bacterial community composition or comparing to other city or suburban site to compare those bacterial community. And most likely source of airborne bacteria in urban site was human. This is already known that in New York, the main bacterial source in New York in winter is a dog skin microbiome. So it's very similar result, maybe. And one point of the big point of this firstly, we could detect it Legionella in outdoor air sampling in Japan. So, okay, so from this one, this time we can see the airborne microbes from house. Houses are not airborne, but source of the aerosol in indoor, we can see that microbial composition. Also, we compare the mountain area microbes. It's totally different microbiome, only 40 kilometers difference. And also suburban and urban site is also different microbial composition. It hurts. So each site, each house, or each area has a different microbial composition hurts. So from this, I will briefly show you that the bioresource in Chile. The sampling site is Osorno. Kindly, Osorno, one of my colleagues in Universidad de Los Lagos, investigator, gave us the filter from the Osorno sample. And Osorno, as you already know, that already one of the really polluted region in Latin America. This air pollution in Osorno, especially higher in fall and the winter. At this moment, at this month, July and August, it's very high. So they give us the air filter at this moment. And mainly this highly pollution cause is a PM 2.5 emission from industry or car or burning. It's very crowded with this PM 2.5 particles. The sampling city is, they collected using this truck and portable sampling machine and go to the Lagos is here, Los Lagos is here, and the El Alba station is here. They collected sample moving this point, all of this point. They collected the sample driving the car. This is a portable air sampler. The putting on here the filter, and they collected about 3 to 4 times per week. And the sampling is one spot is 15 minutes, and 2 liters per minute, and 13 liters per spot they collected. So total air volume is 450 to 600 liters of the air they collected. And thanks for them, I bring back these filters to Japan and extract the DNA. And just extracting the DNA, the difference is already occurred. This is a negative control. The total amount of DNA is like this. And pre-emergency, alert, emergency, good, regular, it's steadily decrease the DNA, amount of DNA. So maybe pre-emergency is a really high contaminated air in the microorganisms. And we analyze the microbial composition, and we know that we found that this is a bacterial composition, hierarchical clustering. This is a 18S eukaryotic microbial composition, comparing to clear air and high polluted air. This shows that the highly polluted area and the clean air, this 0 is a blank one, is separated microbial composition. 18S1 also showed a similar result. This is a blank, so clear air here. So clearly separated this one. So maybe we can detect, we can use some indicator microorganisms for high polluted area or clean area. This is a result of 18S composition. Major is a mammalian. So maybe human or dogs and the cat. We don't at this moment which one is true, but the main is mammalian. And the other one is this is animal parasitic yeast. Is the second one, and third one, and the fourth one is a mushroom one. Looking at this microbial composition, we can microbial, not eukaryotic composition, we can not know the difference, but seeing the hierarchical clustering, it's a clearly show that maybe we can continue sampling and we can detect some specific eukaryotic indicators. This is a 16S bacterial or prokaryotic bacterial composition. Major is Ralstonia. It's a soil bacteria and some plant pathogenic bacteria. The second is Staphylococcus. This also including a pathogenic species. And the third is non-assigned one. So maybe I'm not sure what new microbes in there. Or fourth is human skin and mucosal surface associated bacteria. And the fifth is Pseudomonas. This one is everywhere. So it's natural or natural result. This also Streptococcus is including a pathogenic species. So a lot of pathogenic species inside the microbial bacterial composition. So we keep sampling, and this is very, we need to check this is very pathogenic species or not. We keep sampling. And also we sampling at the Universidad de la Frontera. It's in Temuco, one of the polluted city also. Using this high volume air sample, and this is a Coriolis air sample. One is for bacterial community or microbial community using this sample and this sample. The other one using Coriolis sample. Some sample is used for culture analysis. At this moment, we have 23 strains from Temuco samples. We are looking for to analyze what kind of bacteria here. And also we are sampling Punta Arenas. It's a clearlist regional city, and also we sampling at the Universidad de Magallanes, high volume sample, and Coriolis also. And at the Coriolis, from the Coriolis sample, we can get 20 strains using aerosol sample each other. You can use Japanese aerosol samples, and also we can use Chilean aerosol samples. So we are looking for that this MTA sign in the future, near the future. And this is almost the final slide. This presentation, this is just a commercial for the SATREPS project. We are now invented that suitcase lab. What is a suitcase lab is from sampling for seawater, sampling to detect the specific bacteria or algae microorganisms. We can detect it on site. The procedure is like this, sampling and filtration, and at the home or hotel or in the car, you can use this one. And using this one, you can detect the specific, using a lamp primer, lamp method, you can detect the specific microorganism. So I think we can use for other like viruses detection or bacteria detection. We can use we can use for this one. And we also invent next version suitcase lab. It's more thinner and more lighter. So if we complete to finish this innovation, I want to bring this for your country. Okay, thank you very much.
Okay, thank you very much. So, for your wonderful talk, it was very interesting colloquium. We have a lot of participants here from different parts. So, the colloquium is open for questions. Se abre a la discusión, al público. Vanessa, no sé si están habilitados para preguntar.
Sí. Daniel
Valenzuela. Daniel, por favor.
Hola.
Please on English, if possible on English.
Good night. Thank. I think a question. Well, we know that these bacteria are active individually, but I know if the activity is social and social interaction between different tags in the airborne particle. This is the question.
Asking about the social interaction of these different bacteria or fungi in the airborne samples.
If the activity is social, that the tags interact between them and transfer genes, metabolic product, or nutrient on the airborne particle.
I see. Maybe they interact with each other, maybe, but we don't have a result at this moment. But it's very interesting point of view. I want to know that about that.
Thanks.
¿Más preguntas? So for me, it was very interesting that you was able to detect in the air of one of the cities Legionella, no? Because you say the first time that Legionella was detected in the air, and I think, of course, the air, what you have shown with your studies is that the air is a very important source of infection, of different infections. And about this Legionella, but which could be the origin of this Legionella in the air in this specific city, but not in other cities of Japan?
Yeah, maybe, yeah, Toyama is especially a lot of people infected by Legionella, and Toyama Institute people said that the main source of Regeneria is a pond, or water pond.
Okay.
So after the rain, the Regeneria infected people are increasing, they said.
Yes, but what is different from this city to other cities that also have these water ponds after the rain?
Yeah. So, why?
Yes.
It's my thinking, just my thinking. But Toyama University, not Toyama University, Toyama area is surrounded by mountain, as you see in the map. So Regeneria in the water is come from the mountain to city area, and maybe stay there and evaporated with aerosol as aerosol and infected. Maybe the main source is a mountain water. I'm not sure.
Okay, thank you very much. Now Francisco Cereceda wants to make a question. Francisco?
Hola, hola. Hi, so hello. Thank you very much for your presentation, and so. Thank you. One question is, what will be the most common origin of the microorganisms that you have found in the mountain areas? It is from local or from long distance transport?
In our result, it's maybe a local one. So they live in the mountain, and they sampled or bloomed in the wind or collected in the sampler.
And then you have the similar origin that you find in Temuco, from animals, from mammals, from humans, and or from another source?
Yeah, we want to analyze the Temuco sample, but we are waiting for MDA aside. So we don't have that data. Oh, I think soon we can get that data.
But you can expect it maybe also microorganisms from another part of the continent, maybe in Korea, or is it only for local from Japan?
Yeah, Toyama is, Toyama can get the Chinese desert not soil, in the China desert, the wind blown. So they always affected by this phenomenon. So maybe some bacteria is come from China, or some study already show that data come from China.
And you can so identify the origin from these microorganisms. Then is it so specific that you can identify from microorganisms from China or from Korea or from Japan?
It's it's very difficult. The microbial origin. So we need to analyze that data using a model, some wind direction model, and not as well as the wind direction model. I think we need some isotope traceability tracing. We also need that and create that what does it say?
Yes, you can
this
combine.
Idea.
Yes, this is a different method to identify the source. Maybe this high split trajectory of the modeling and of the in this case the microorganisms fingerprint, the chemical that are combined together to identify the origin of these microorganisms. Thank you. Thank you so.
Ok, ¿está abierto a preguntas? No sé si alguien más ha levantado la mano.
Sí, yo la levanté. No sé si puedo hacer una pregunta.
Por supuesto.
Hello, thank you very much for your presentation. It was very interesting. I have really enjoyed it, and my compliments for you. Oh, well, I didn't know that Osorno was of the most polluted city of the world. So maybe the particular matter are completely different in all the long of Chile. My first question is related if you have observed a relationship between the bacteria community or the abundance of the bacteria community with polluted sites or another kind of aerosols. For example, here in the north of Chile, the aerosols are coming from the mining industry, so it's completely different to the south, where is the source of the particular matter is coming from mainly from combustion. So have you observed a relationship between all this diversity of the community between the pollution condition?
Only the polluted condition is Osorno data, and the Osorno data show that polluted one and the Korea one is totally different, but we need more data. So I cannot say at this moment clearly it's different. I cannot say that, but maybe it's different. So especially with soil, a lot of microbes are attached with soil. So maybe or probably it's different, and very interesting that same pollution but different source. It's very interesting point.
Thank you.
Gracias, doctor Luis Rojas, desde Antofagasta. Ahí está abierto todavía el espacio para más preguntas. So, I have another question, because you were speaking about Mycobacterium. That is not related with tuberculosis directly, but you can find it in the bathrooms in Japan, no? And so, what is the problem with this Mycobacterium? Which is this also an animal pathogen, or?
This one also animal pathogen, the human pathogen. It's this human and animal both infected pathogen, and for human, especially in Japan, there's a lot of Mycobacterium infection is now increasing. The Mycobacterium is very no Mycobacterium NTM is difficult to treat. It takes a long time, and sometimes it's very hard infection to human. So it's very important for human health, especially Japanese elderly people.
Okay, it's there. Yes, and you know if the same happen in other cities in Asia, for example in Korea or in Singapore or in China or I don't know, Vietnam.
Yeah, in Korea, it's the same type of NTM. The Japan is occur, and also there is not so much data in Indonesia, but also Mycobacterium is very problem in Indonesia. So we are studying at this area of country.
Okay, no, that's very, very interesting. So we are very happy that we have connections from different countries. You're not the only one connected from outside of Chile, so that's a good news. And in all our colloquia we have had people from other countries. So I offer again. ¿Hay posibilidad de hacer más preguntas? Sí, Miriam, perdón. Miriam.
Acá estoy.
Sí, que tú levantaste la mano.
Sí. Hello. Thank you for your presentation. It was very interesting. For me, it was very interesting because you found Rastonia in the air of south of Chile. I work with plant pathogens, so it's very interesting. And I would like to ask you if it's possible to identify the species of Rastonia.
So if you have a primer for species analysis, we can detect that. This sample is only short reads with data sequence. So if you have any specific primer to detect the species for Rastonia, we can analyze that.
Yes, we have.
So
because you have the DNA.
Yes.
And
yes,
only the strain or only the DNA?
Only DNA we have.
Okay.
Good. So let me let me teach the specific primer information so we can detect that Rastonia information.
Yes, so surely Miriam can send you this information because we reported this year for the first time in Chile that the Rastonia is affecting some plants. It's a very important plant disease in Chile, so we're very interested in this, and perhaps Miriam can come in contact with you and we can see if this is possible to identify.
Yes, because in the south of Chile it's a quarantine disease. So it's important to know if it's really Rastonia solanacearum or not. Thank you.
Yeah, thank you too.
Okay, so is there any other question? Vanessa, perhaps you have some information that you want to share?
I just want to say thank you to Saul. It was a great presentation, and thank you again.
Thank you too.
I want to, okay, bueno, so thank you very, very much. We're very happy that you are still awake and giving us light about air microbiology, because in Chile we don't have much experience with air microbiology. We have less experience in these than other countries like, for example, Puerto Rico or Cuba. They have done much more studies about air microbes than us. So this is something that we have to do as a country. You know? Therefore, the help of you and of Fumito could be very, very important. And I also want to say to the people that we will have also next month another colloquia. Professor Sara Cuadros from the Universidad Católica de Talca Universidad Católica del Maule, but of the city of Talca, will give a very interesting talk. So you are all invited to share this very nice and high level colloquium of Valparaíso. And I want to thanks again. So was a very great and inspiring colloquium. And we hope to see you and to see Fumito soon also in Chile. So you can give another talk, but we can have more social contact between us. Okay, so to all the people, goodbye and thank you for sharing this colloquium.